Hello all, welcome to F.Y.B.Sc class. Today we are going to discuss about cultivation of microorganisms from the unit Microbial Cultivation and Growth of F.Y.B.Sc syllabus paper 2 MB122 Microbial Cultivation and Growth. The learning objectives of today's lecture is to understand different methods of cultivation of different kinds of bacteria, like how to cultivate photosynthetic bacteria, which medium we can use, what are the methodology for the cultivation of such a kind of the bacteria. In addition to that, we are also going to discuss how we can cultivate or isolate extremophilic bacteria and cultivation methods of chemolithotrophic bacteria. Now we are going to discuss about the cultivation methods of photosynthetic bacteria. So here we are taking the examples of strains of purple non-sulfur photosynthetic bacteria, Rhodobacter spheroides, species G8 and species A10. Now we are going to discuss about the cultivation method related with these two bacteria. So these two bacteria were isolated from the mineral spring of the Jermuk and the Arzini. In addition to that, Bacillus megaterium species has been also studied. Now, after their identifications, all these strains were deposited in culture collection of the Center of Microbiology and Microbial Depository, NAS of Armenia. So, these strains of phototropic bacteria were first grown on the Ormerud's medium, while the strain of Bacillus megaterium was grown on a medium which consists of fish hydrolysate. For the cultivation of photosynthetic bacteria, we can make the use of agricultural waste like the manure and poultry dung can also be used for the cultivation of photosynthetic bacteria. Now, this wastage which was collected, they were first dried at 60 degrees Celsius for 15 days. Now, this wastage should retain the humidity. In case of the manure, it should retain 30% of humidity, while in case of the dung, it should retain 20% of humidity. Now, after their drying, the 100 gram of this dry matter sample, they were dissolved in 1 liter of distilled water and the solution were kept under room condition with periodic stirring for 4 to 6 hours. Now, extract was obtained by the filtration of above solution, and that solution or the extract we can use as a base of nutrient media for the cultivation of photoautotropic bacteria. Now, the pH which was maintained, it is in the range of 6.5 to 6.9. So, the chemical analysis of the extract, that is the solution, it will consist of ions of bicarbonate, then carbohydrates in the form of glucose, sucrose, salts of organic acid. Now, all these are media is designing in such a way that it will satisfy the growth requirements of purple non-sulfur bacteria. Within this medium, the organisms will be grown at 30 to 32 degrees Celsius with 2500 to 3000 lux illumination under different conditions of the aerations. Now within this we have added the inoculum, that is the microorganisms, in amount of 5%, and the growth of the organism was monitored either by using spectrophotometric analysis, where we go for the determination of optical density of cell suspension at the wavelength of 660 nanometer. In addition to that, we can also monitor the growth of culture by determining the dry weight of bacterial biomass. In addition to that, the microbiological analysis was performed by using phase contrast device. So, adsorption spectra of all whole cell will be measured on UV spectrophotometer. For the growth of these photoautotrophs, they require 5 to 8 days. The protein content of this organism was monitored by using nitrogen method. And by using amino acid analyzer, we can determine the amino acid composition of photoautotropic bacteria. Next is to study the cultivation methods of extremophilic bacteria. Extremophiles are the organisms that thrive or survive in extreme environments. We can define extremophiles as the organisms that live in extreme environmental conditions like the high pressure as well as high temperature. Examples of such a kind of the bacteria are those bacteria which are found on the rocks near the hydrothermal vents. Here we are going to discuss about the cultivation of extremophilic bacteria in concern with the cultivation of nalgus bacteria. For that, we are designing the inorganic culture media, which mainly consists of hydrogen, oxygen and the CO2. Now let's discuss the isolation methods of extremophilic bacteria. The major drawback of this extremophilic bacteria is that some of the thermophilic Naegle's bacteria which grow but they will not form the colonies on the agar medium. We can take the example of hydrogenobacter which do not grow on solid media with only hydrogen as an electron source. So, to cultivate such a kind of the hydrogenobacter strains, we have to design the medium which should consist of thiosulfate under hydrogen which consists of gas mixture or under air. Then only it can form the colonies. Now, if growth does not occur on the solid medium, we have to go for the repeated dilution procedure so that to obtain the good culture of the organism. Now, here we have added 1 ml of inoculum within the design medium. So, to obtain the most representative or the most interested strain from the given environment, we can go for the serial dilution method of the particular sample so that it will reduces the microbial load and that each dilution we can use for the enrichment culture. Now, the last dilution was used for the further isolation process. In case of the extremophilic bacteria, we have to give some pre-treatment to the sample. For example, boiling of the sample for 5 minutes so that we can select for the spore formers. Next step is the enrichment. The enrichment methods or the procedures of extremophilic bacteria, they are more simpler because these are the only some of the bacteria which are able to grow under that specified conditions. So enrichments will be carried out in liquid culture, and for that the medium will be dispensed or added in a shallow layer so that adequate exchange of gases should be there. Now, to obtain the pure culture, enrichment process will be repeated for the several times. The medium which we are using for the cultivation of extremophilic bacteria are the basal medium. So while designing the medium for the cultivation, we must take into the consideration the required growth factor for that particular bacterial cell. So in case of the nalgus bacteria, while designing the basal medium, inorganic medium should consist of essential minerals like the potassium, magnesium, calcium, sulfate, phosphate, iron with ammonium source. So this provides all the essential growth factor which are required for the growth of that organism to be take place. In addition to that, this media should consist of high concentration of phosphate. Now, because of the it is necessary to maintain the neutral pH, but because of the phosphate, high concentration of phosphate, it will be difficult. Now, if you are interested in cultivation of thermophilic bacteria, then you have to cultivate or preserve them or isolate them at high temperature or at elevated temperature. Now, to obtain the pure culture, to maintain the growth of that bacteria, we have to stop the incubation after the stationary phase is reached, and you can preserve your culture to refrigerator temperatures. Now, what are the incubation conditions for the cultivation of extremophilic bacteria? The gas mixture which we are using for the isolation of Naglus bacteria, it should retain a reduced amount of oxygen, as some of the bacteria are microaerophilic or in lithoautotrophic condition. That is, they require very small amount of oxygen to carry out their growth and metabolic activities. So, while selecting the gas mixture, we should design or select the gas mixture which should consist of 0.03 to 0.05 or zero point 0.1 bar of CO2 gas and 0.75 to 0.85 bar of hydrogen gas. Now, The oxygen content within a particular medium can be increased stepwise to obtain the higher cell number or the cell density. Now, for the cultivation of this bacteria in the petri dishes or small liquid volume, we can also place our culture in the desiccator. First, the evacuation of all the gases which are present within that desiccator or the Witts-Weisser should be carried out, and then we can add the desired gas mixture. Now, if the incubation conditions are such that it requires high temperature under pressure, then such a kind of the high temperature condition can be provided by using the hot air oven. Now we are going to discuss about the cultivation of chemolithotrophic bacteria. So chemolithotrophic bacteria, here we are going to discuss about the enrichment of those bacteria which require or which will grow in presence of high concentration of nitrite. Examples of such a kind of the bacteria are Nitrospina gracilis. Now, if we are cultivating the chemolithotrophic bacteria which grow in a higher concentration of nitrite, then Nitrobacter will be the dominant species within that culture. Now, in case of the chemolithotrophic bacteria, it is important to use low as well as high concentration of nitrite in enrichment culture. Now, the source which we are using for the enrichment culture will be from ammonia oxidizing bacteria. Nitrite enriched medium. First, we will carry out the dilution of a sample to obtain the dominant and the fastest growing organism. Now, the enrichment cultures, they are incubated in the dark at 25 to 30 degrees Celsius for 1 to 4 months, and we have to frequently examine for the oxidation of nitrite. We can also perform the spot plate analysis method where we can check whether the oxidation of the nitrite within the media is take place or not. Now, after the serial dilution of the cultures and after obtaining the appropriate growth, we can transfer the growth to the fresh medium where we have added additional nitrite so we can obtain the more pure culture. Now, to obtain the turbid culture, we can periodically add small amount of nitrite into the medium. Once you will get the turbid culture, then we can go for the methods of obtaining pure culture by using serial dilution or the plating techniques. The nitrite oxidizing bacteria, they are having a prolonged generation or the doubling time which is 8 hours or more. In some of the cases, it may require several months to obtain pure cultures. These are the references. Thank you.